Good morning, everyone. We'll be starting in 10 minutes. Good morning, everyone. We are starting our conference. Good morning, everyone. We welcome all of you to the second day of the International Conference on Innovations in Biotechnology and Life Sciences. My name is Japneet Singh, and on behalf of the ICIBLS committee, I welcome you all today. Today, we are geared up for some exciting talks. I am overwhelmed to announce our first speaker for day two, Dr. Monideepa Roy, who is currently the Vice President of Akamara Therapeutics. Her first entrepreneurial endeavor was to establish and lead the R&D laboratory at Invictus Oncology in New Delhi. There she led a team of accomplished scientists to develop a pipeline of novel supramolecular anti-cancerous compounds. She was also responsible for creating Akamara Therapeutics as an US entity in Boston. After being doctorate from GNU Delhi, she was trained in the Department of Pathology, Brigham and Women's Hospital, and received a fellowship from the US Department of Defense. Her innovative work was honored with the Thomas Gill Prize for Research Excellence at Brigham and Women's Hospital, Boston. She is an inventor on several international patents. Now, I am very honored to welcome Dr. Monideepa Roy. Over to you, ma'am.
Thank you very much for the very kind introduction. Am I audible?
Yes, ma'am.
Yeah, so I'm going to try and share screen and just to make sure that it's going to work. Let me see. Is my screen visible?
Yes, ma'am. Yes, ma'am.
Okay. So I would like to thank ICIBLS organizers and Dr. Asmita Das for the kind invitation. It is my honor and my privilege to be here today speaking to such a distinguished audience. I also always love to interact with young minds, and I'm really very happy to be here. So I decided that, I will utilize the work that we have done at Invictus and taken it forward to walk you through the roadmap of what we try to do using the ecosystem of innovation in India. It was very inspiring to hear Professor Bhatnagar and Professor Singh yesterday talking about innovation, particularly such impressionable minds as the DTU students and other students who have joined the conference. And I think India is on the right path of harnessing all of that competence and all of that willingfulness to take innovation forward. So without further ado, I want you to look at this particular number. Are you able to see? Yeah.
Yes.
2.5 million. That is the number of cancer patients living in India today, 2020. It's a huge tumor burden. The healthcare system that we have in place is not accessible by everyone. It is a tremendous issue that we all need to be paying attention to. In fact, just to give you an idea, by the time I will finish my talk today, the global number is actually even more staggering. By the time I finish my talk, across the world, 350 people or more would have died of cancer. So, I like to call cancer patients in a chronic type of disease management situation. It is a situation where they live with cancer. When we hear the diagnosis cancer, it's almost like a death sentence, right? But because of technology, because of innovation, because of your minds and people's intent to come up with more and more innovative solutions, It is possible to manage many, many cancers and many, many cancer patients' treatment today, just like diabetes or cardiovascular disease. But this cost for living with cancer is tremendous. There are lots and lots of new drugs that are coming up on a daily basis. There were this was a COVID year, so I would say the number of approvals may not be as high as other years, but 60, 70 drugs are approved on a yearly basis by the FDA, more than 20 of which is in the oncology space. But if you look at the numbers at the very bottom of the slide, it's overwhelming. The economic impact of the therapies is overwhelming. So although the therapies are out there, they're not accessible by all the patients. There is another challenge that I'm going to walk you through. In India, most of the treatments that are available and that are given to the patients are approximated based on data that we obtain from clinical trials. And these clinical trials are done or conducted largely on the Caucasian population. Over the years, there are Asian data coming through, but again, those are mostly from the Far East. There are some data from India, but nearly not enough. So the big question is that the data that we get out of these clinical trials, is that even effective considering the genetic diversity that exists in our country? I have one small example on the right-hand side here, which is labeled as the India puzzle. This is just breast cancer. If you look at the comparison, the average age is different, affecting younger women, lower than 40. That number is very high. The grade is at diagnosis, is a higher stage compared to Caucasians. The percentage of tumors that remain refractory to therapy, that's about 10% fold higher. So the therapies that are out there, they need to be extrapolated in a way which is driven by science, which is driven by understanding what the genetic pool is out here in our country. So a lot of work needs to be done in this area. Now, making all of these therapies accessible or digging into such informatics to make sure that we are able to manage cancer as a chronic disease, big dollars need to be spent. I mean, without spending the money, without the investment there, how can you take research forward? Now, here's a snapshot of a global R&D landscape. This is 2020. Now, just to be able to understand this map, the diameter of each circle is basically the amount in dollars in spending. So you can see United States, the diameter of the circle is pretty big. So is China's actually, So the R&D dollars spent much higher in USA and China compared to India. Now, on the x-axis of this graph, you can see R&D spent as a percent of GDP. Now, most of these countries, United States, Germany, Japan, Sweden, South Korea, they are spending 3 to 4% of their GDP in R&D. On the other side, if you look at India, it is clubbed with all the other developing countries. We spend less than 1% of our GDP in research. Now, I'm not going to, downscale what is already there. It has increased from 10 years back, but there has to be collective thinking as to what is important. I'm not saying that putting a lot of money right into the research without understanding what is actually required is going to be beneficial, but a lot of people need to come together to figure out the solution. If you look at the y-axis now, this is where all of you come into play. This is the number of researchers per million population. And you can see where the developed countries are, and you can see where India is, right at the baseline. So we need more people to do the research, to carry out the innovation, innovate how to carry out the innovation, and figure out the policies of the country. So how can we make a difference? If you look at the left-hand side here, the data is from 2018, which you can sort of extrapolate to today. So this is the current scenario where the India pharma industry the size is at 38 billion US dollars. Now, if you think 10 years ahead, 2030, whatever we are going to do, we are going to develop further. So you can imagine that there would be doubling of that size, right? But is there a possibility to increase this base case to a really nice growth, very accelerated growth, where not only disease like cancer would be taken care of, but also other diseases, diabetes, cardiovascular, infectious diseases, all of this can be addressed? What are these key drivers that can help us achieve more? So one thing that we have to think about is that we have to make healthcare accessible by all. Right now, we have only less than about 30 skilled health workers for every 10,000 people in our country. If you compare that to US, that is more than 100. Now, we cannot jump to more than 100 in 10 years, but can we take this number from 30 to 50 to 60? Why do we need to do this? Because if we can make healthcare accessible by most, then the disease burden reduces. You can bring in screening procedures, you can bring in prevention procedures, and then the overall burden decreases. So we are talking about innovation. That is the benchmark, that is the foundation, but there has to be attention given to other things, such as making healthcare accessible to all. We talked about innovation here yesterday. It was so nice, so inspiring to hear from Professor Bhatnagar about innovation, about how insulin made a difference. We have to carry that effort forward. We cannot stop where we made better insulin at a lower price. We have to figure out what are the newer product classes, what are the biosimilars, what is the gene therapy that's available there, what are the specialty drugs. And having driven innovation with a small team in our country, one thing that I've understood, we really need a lot of cooperation from the policymakers, from the ministries, from the regulatory boards. I remember that we had compounds ready in hand 10 years ago. It took us a year and a half to get an animal facility in place to test these compounds. So such bottlenecks, these need to be removed, or they need to be streamlined. They are streamlined now. These days, if you have to apply for an animal facility, it's an online process. So much easier than having to go to the Ministry of Environment and Forest, like several times in a month to follow up on your case. So things are improving, but more focus needs to be given to make it a key driver of growth. When we wanted to manufacture our drug, we have the technology, we have done the innovation, we have thought out of the box. We didn't have the supply of the starting material. Where did we get the starting material from? We got it from China. Why did we have to get it from China? It was unaffordable, what we had in India. How can we become the world's largest and most reliable drug supplier? That is something that we have to think about. We have to have the standard qualities in place so that we can manufacture at that level to support innovation. And then we have an excellent workforce. We have more than 225,000 pharmacy students graduating from our education ecosystem every year. Compare that to US, 17,000. Can you imagine the size of this workforce? We have to be able to leverage that. We have to find out what students want to achieve in life. Even the basic research that we do, I was so impressed with Dr. Mithilesh Mishra's work yesterday, discussed yesterday. It was amazing to see, what happens in cell division, how the actomyosin motors work. Can that be understood in a disease scenario also? When Röntgen discovered X-rays 200 years ago, he was not thinking that he's making an imaging technology to be used by one and all 200 years down the line. But that was basic research, and we cannot imagine our lives without X-ray today. So basic research and translational research need to go hand in hand. We need to be able to understand problems, and we need to be able to apply that understanding in fighting disease. Lastly, one more important point: many drugs are going off patent by 2024 of the scale of 250 billion US dollars. If our pharma industry can develop a molecule by molecule level strategy, and then put the regulatory and market decisions in place, market excellence in place, nobody can stop us. So these are just some of the learnings that we have understood over the years in our innovation process. And I wanted to share that with you because science is one thing where there's a lot of career opportunities. But, at least when I was, learning about science, I was not that aware about all of this support, understanding that's required to drive an innovation forward, to drive technology forward, and therefore to drive our country forward. So people who work with cancer are very, very familiar with this Hanahan-Weinberg model of hallmarks of cancer, basically how the cell aberrations can, signaling aberrations can lead to growth in, altered growth in cells. Similar to this hallmarks of cancer wheel, there is a hallmark of technology that can help us manage cancer. So there are so many types of technology that is out there, from the therapeutics aspect, DNA editing, vaccine, cell therapy, biologics, small molecules. Also, let's not forget the imaging, the AI part of it. Now, it's very difficult for one organization to do everything at one go, right? Particularly for a country like ours, we should think about pooling resources across the centers of excellence. And, we have been privileged by being in Delhi, being around institutes such as Delhi Technological University, such as All India Institute of Medical Sciences, being in close crosstalk with Indian Institute of Science, Bangalore. So the model that we have discussed with the clinicians, with the bioinformaticians, with the researchers, is that if there is a patient workup that is happening, keeping the patient privacy in place, if we can get the data that can be processed, the molecular markers can be understood to follow the progression of disease. IGIB has such a wonderful world-class tissue repository. Whether we can mine all the data that's there, use bioinformatics, our country is so great in IT. We can understand big data, have all of this learning in real time and bring it back to the clinician for decision making. These are some of the thoughts we put together, we learn together with industry and academia. And some of these, I'm very proud to say that our hospitals and centers of excellence have taken up. And the decision making in the clinic is progressing forward. So this is an example of how it can be a key driver of technology. Now the story of Akamara Therapeutics. So we started out with our vision of reimagining immunotherapy, and we wanted to move treatment beyond the limits of history. Now, how we would do that? We came forward with an idea that we are going to use the humoral system, B-cell therapeutics, to change the way people live. That was our mission. And for that, we aimed to innovate and develop paradigm-changing therapies that would impact patients. So largely our R&D unit is based in India. It is Invictus Oncology. It started out as Invictus Oncology and later on became Akamara Biomedicine. And the headquarters is in the US. And here also, one point is there that some of the regulatory paths are well defined in the US setup. And so if, our regulatory paths became a little bit easier to navigate, some things, can be driven from India as well. So, the conventional approach to take care of cancer has been get out all the weapons of mass destruction that you have and go, and slam the disease cells, and then collateral damage are the healthy cells, like, attack all the cells that you have. And thereby, what happens is there's debilitating side effects, putting healthy tissues at risk. There's, we all know about hair loss or, pain, rashes, anemia, cognitive changes that are associated with these quote unquote weapons of mass destruction, namely surgery or radiation therapy or chemotherapy or even immunotherapy. So can we imagine, using a postal address, a postal stamp, where we could take all of these weapons and, figure out pinpoint the place, pinpoint the target, where it is going to go and exerts its effect, much like, enemy missile launching, and not affect the healthy tissues where the civilians live, right? Whether that is possible. So I'm going to walk you through two different technologies today. We have worked on several. First, I'm going to talk about an antibody drug conjugate. Now, the magic bullet is a concept that was developed more than 100 years ago. And Paul Ehrlich coined it. He even won the Nobel Prize. Of course, he was not trying to treat cancer at the time. He was using the concept of magic bullet to treat infections. This was prior to Alexander Fleming's penicillin. So Paul Ehrlich thought that there is a way to take care of disease cells and not harm the normal cells. So extrapolating this magic bullet concept into cancer, if we are able to find the address of the tumor cells, use a postal stamp, and deliver the drug to that address, can we do that? So 2-3 decades earlier, the seek and destroy model antibody drug conjugates were conceptualized. Basically, you have an antibody that can go and target a protein that is exclusively expressed on a tumor cell and not so much on a normal cell. Now, one would say, well, therapeutic antibodies are already there, so why not just use that? Or why not use a small molecule that goes and blocks the receptor that's also targeted? There are issues with small molecules. They're small. It can go everywhere. Even if the target is not expressed, they are, they can metabolize in a way, affect the cells, affect the healthy cells there as well. With the antibodies, yes, they are going to go to the cells that express the antigen. Problem is, they don't have that great cell killing capacity. So it was conceptualized that perhaps we can empower these antibodies with a payload, which would be a chemotherapy drug that can kill tumor cells. Now, the difficulty was that you wouldn't want that payload to release in circulation, because then you have that problem of the payload going everywhere. So you had to use a linker that would remain stable in circulation, but once inside the tumor cell, would release the drug, and that would kill the tumor cell, sparing the normal cells. So you'll get more efficacy and less adverse effects. Problem is there with the target, with the payload, but we felt that the greatest problem was there with the linker. Now, why do I say that? So the linker can fall off in circulation. If you make it too stable, it can not release the drug. Now, also, if you are going to attach a linker and a payload, perhaps the antibody is not going to recognize its antigen. There are so many issues with the linker. So what we thought that let's think of building blocks. Antibody is a building block. Linker is, several atoms of some other building blocks. And then you have a payload, which is the third building block. So we did our in silico computations of bringing in 2 or 3 different components of these together and creating these ADCs. And the problem with the conventional ADCs was that they would attach the payloads to a lysine or a cysteine residue. They would get different numbers of drugs attached to antibodies, giving rise to very heterogeneous ADCs. That means depending on what type of ADC is reaching which type of cell, the effect would be very difficult to monitor. So for that, to troubleshoot that, the lysines or cysteines would be engineered in a way that only site-specific payloads would bind, giving homogeneous antibodies, homogeneous ADCs. Problem was engineering such antibodies was really costly. So what we thought is that we are going to use a linker um, uh, it's a markup to ethylpyridine MEP linker, which is used in purifying antibodies. So it binds very strongly with the antibody, but once inside the cell, inside the lysosome, the acidic nature would make it release the payload. So we used this linker, we modified it to have our payload, and we found 6 specific and conserved binding sites. So always our drug antibody ratio would be 6, therefore we would get homogeneous ADCs. They would be very stable conjugates. And the technique was very simple. You put the 2 things in a tube, it would conjugate. So no modification of antibody was required. It was defined sites, low heterogeneity. The higher order structure was preserved, very stable. The main selling point was it took less than 2 hours to process. So you can imagine now you don't even have to think only about a particular cancer that is being targeted by a particular antibody. It can be different indications. You just need to know the target and you have to have an antibody against it. And you can use the payload and the linker, and in situ, just before injection, you can put them together and inject it. So that was the innovation. And we found that, so here on the left-hand side, T47D is a cell line that does not express HER2 receptor. SCOV3, this is a breast cell line. This is SCOV3 is an ovarian cancer cell line that expresses the HER2 receptor. And we made an HER2 ADC, and in the payload, we used a fluorescence dye. And you can see that where the cells have the protein expressed that recognizes the antibody, the ADC penetrates. And where the receptor is not there, it does not. And similarly, when we looked at the cell viability of these cells, so instead of using a fluorescent tag, when we put the actual payload, the gemcitabine drug is what we used, we found that wherever the receptor expression is high, the cell killing was very, very efficient. Wherever the receptors were low, the cell killing was non-existent. So we had achieved specificity. We, of course, extrapolated that to mouse models. This is a lung cancer mouse model. On the y-axis, you see tumor volume. The black line shows the tumor growth without any intervention. The green line shows with just a regular antibody that does not have that great a cell killing property. And the purple line shows our ADC, which really flatlines the tumor. Considering mouse tumors, the effect is very long lasting. And there is no loss in body weight, which is very important because if there is toxicity, then there would be decrease in body weight. So we got the survival benefit. The purple bar here shows increased apoptosis, which we scored with tunnel assay. And we also looked at certain parameters of toxicity, such as the blood cells and the liver toxicity and the kidney toxicity, and we were not able to record any measurable toxicity. So it gave us the ADC that very easily manufactured and has a great effect in lung cancer model. So this was an adaptable platform. You can use any antibody, you can drug whatever has been so far, quote unquote, undruggable, because whatever your target, you can make an antibody against it and make an ADC of it. It's very versatile. You can make it a universal one affecting many kinds of patients. You can make it personalized depending upon what kind of mutations are expressed. And it can be used across therapeutic areas. It is not restricted to cancer alone. So we published this work here, again, the regulatory path for taking this into clinical trials was way too much for a small startup biotech like ours. So we have published this work, and our goal is to license this technology out to established organizations that can take it forward. Now I want to switch over to another technology, and here I am not going to be able to share too much detailed data because this is our lead asset. So right now, immunotherapy in cancer is about not attacking, but modulating T cells in a way that the immune system can fight cancer. And there are already 700 plus companies that have various kinds of technologies that can activate T cells. Now we thought that, this is immune response. T cells cannot work alone. There has to be other immune cells. We already know that macrophages and natural killer cells, have a big role to play. But there was controversy about what role B cells would have to play in cancer therapy. So we thought that we are going to harness the power of humoral immunity and bring the promise of adaptive immunity into how we manage cancer. So I'm going to talk about our lead program, which is now all written up for what is known as IND filing, which is investigational new drug application, based on which the regulatory authority would give approval for running human clinical trials. So we have very nice preclinical data where we see tumor regression in several types of tumor models with very limited toxicity. This is a completely unique mechanism of action, and we have IP exclusivity until 2033. It has a novel mechanism of action, and it induces immune memory response. And the idea is that, the T cell immunotherapies have, revolutionized the way we approach cancer therapy now. But there is still a high group of high subset of patients who are refractory to these approaches. So we believe that, where the T cell therapies are not able to create an impact, our therapy would be able to, arrest tumor growth. So it could be used as a single agent or in combination with a checkpoint inhibitor. Now, in certain tumors, already literature shows that B cell response is associated with long-term survival. And this has been mapped by one marker, which is known as immunoglobulin kappa chain. And it has been seen that when this marker is expressed highly, there is long-term survival of patients. This marker is a marker of plasma cells. These are mature B cells that produce antibodies. So this is our typical tumor regression profile. On top, you see triple negative breast cancer. The blue line is our drug. And when we do a profiling of our treated tumors, the biggest overshoot of expression of marker that we see is that of IGKC, just as seen by other reports, other clinical trial reports. Similarly, this is the second one here is lung cancer. And here also we see a big induction of IGKC. I can only share data that is there in the public domain. We have, all the data accumulated that, shows the mechanism of action, what the target is, how the B cells are activated. And we are writing that up as a paper, and it should be out, perhaps next year, within the first half of the next year. And then I can share that data. Unfortunately, I'm not allowed to share further today. But long story short, we have finished all of our IND studies. We had an excellent positive US FDA feedback that supports our clinical trial initiation. We have started manufacturing our drug, And we are preparing for the first in human study very soon. Now, our roadmap was not done alone. We reached out to, as you can see on the left side, many academic organizations. DTU is right here. Our students were given the nurturing that's required to keep talent within the organization. And, I will always be grateful to Dr. Asmita Das, for providing a very supportive hand in mentoring our scientists. And of course, various public organizations also, which, enabled us to design targeted therapy for cancer treatment. And I'm just going to end here. This is a slide that I like to share. Basically, our journey is, this is a building that we rented. It used to be a Gutka factory. This is the filling machine of Gutka packets. And that same place we transformed, this is the stage of building it into a lab. Now it's a full-fledged working lab where we do all our R&D work. This is in Patparganj, Delhi. And so very poetic justice that Gutka that caused cancer, now in the same place we are developing therapies against cancer. And with that, I'm going to end. Thank you very much for your attention.
Thank you, ma'am. There are a few questions.
Yes, please.
Will the scenario change if we focus more on improving the living quality than focusing on finding a cure of disease?
You mean lifestyle? Of course. I mean, lifestyle has a great impact. I mean, many of these cancers develop from somatic mutations, which could be, a result of lifestyle. But lifestyle is not the only thing that can cause cancer. It could also be bad luck. So I think we have to take care of lifestyle issues as well as work towards cure. We cannot do one without the other.
And there is one more question. It shouldn't the researchers should work with the clinic because the great research ongoing fails to reach the public.
Yes, absolutely. So I think there should be crosstalk at every level. I was mentioning, right, that, we worked with hospitals and clinicians. Rather, they worked with us. I mean, they are the ones who are doing the hard work every day, managing so many patients, and they are willing to share the data or the tissues in a way that we can mine the data that can help their clinical decision making. So yeah, absolutely, you're right. And that's the way to pool resources and make an impact in the short term.
Thank you, ma'am. Thank you for the knowledgeable talk and your time.
I have a question. Monideepa, it was a very nice talk, and I just wanted to ask something basic about your work. That you had mentioned that you're developing these antibody-mediated therapies, which are also going to crosstalk with the T cells or other cells, I guess. So in that scenario, it's a little different from the ones that you were doing earlier, where you are just directly what do you call it what do you call it you're directly putting the drug in the in conjunction with the antibody and sending it to
antibody drug conjugate, right?
Yeah, but then if over here you are looking at the boosting of the immune response as such, right? So here, what is your experience with the how much long term can you establish that, and which all tumor scenarios you could do that? Because when you're talking about the first approach, you're not really focused with so many interlinked interplay of multiple different immune cells, but here you are trying to achieve that. So what is your experience with that?
Excellent question. And, your expertise shows through your question, of course. So basically, what we thought, so I cannot tell you the target, but basically what's happening here is that we are harnessing the innate immune system. And there is, so you can almost imagine like a viral infection, like a viral mimicry type of scenario. So, when a virus attacks, there would be an interferon signaling pathway triggered. Now, there are components within that system, which is obviously target driven, that is being released from these cells, and that's engaging with the B cells, and then B cell activation is happening. Okay, so this is the entire mechanism, and without being able to explain the target, obviously it's all hand waving. But it's an excellent question, because we thought when our clinician, our chief medical officer was designing the strategies, that we expected a cytokine storm type of side effect, where we thought that, okay, we are activating B cells, who knows what's going to happen downstream, right? So we, in our toxicology studies, for any kind of IND application, you have to do rodent as well as non-rodent model. And we did our rat and we did dog for our case. And we looked for cytokine storm. Our expectation was that there would be a cytokine storm that would be lit up, because it's a viral mimicry type mechanism. We were not able to see that. Now, it's a 6-month study. So it's a 28-day recovery period study. So basically, we'll have to do long term. The 6-month study is something that we have to follow up and see whether long term there is any scenario. Now, at one point you'll have to stop doing the preclinical work. Otherwise, you'll just end up doing preclinical work and you're not going to have any kind of clinical marks. You're just collecting data and making it as best as you can possibly make it. So we have taken a conscious decision, and then the FDA feedback has been very supportive, that some of these things we are going to read out in the clinic. So we are going to start with a very small dose, and we are going to have these readouts, where we'll do panels, and we'll figure out what other effects on what other immune cells will happen. That is one thing, that is from the cytokine level. Second thing that we are also going to do, and also we plan to do at the clinical level, is that we want to do an immune contexture mapping. What do I mean by that? So all the patients would be would be having biopsies. So every time they will go in for their cycle, maybe monthly or whatever, that's something that the clinician will decide. I'm not the clinician and I don't know how invasive it would be, but with those biopsy samples, we are going to do immune contexture mapping. So multi IHC,
Okay.
so for all the immune cells, and that would answer your question as to what's happening.
And
But
another is
yeah, go ahead.
also with the dendritic cells and the macrophages there, that will actually create a little bit of a problem there, I guess, from your point of view. So, I mean, how would you deal that,
Yeah, so the only cytokine to that effect we saw that was a little bit altered was MIP1 alpha, but not to the effect that it was causing any long-term side effects. So that's in our studies so far, what we have with animals, we didn't see it. We expected it. And if we do have that, there are mitigation steps available in the clinic, because even with the checkpoint inhibitors, there are immune toxicities, right? Not necessarily macrophage related or dendritic cell related, but whatever toxicities are there, they are managed in the clinic. So, it's a trade-off at the end of the day.
And there is another question by Dr. Tuhina. She has asked that if the technology can be used for infectious diseases.
Yes, that's exactly what I was saying, that yes, not the one that I talked, not our lead asset, because that is very cancer specific, the way the target happens. But the other one, the antibody drug conjugate, absolutely. And there are nature papers out there for ADCs in infectious disease. I will not be able to recall it right at this moment. But that's the thing, when I said that it can be used in any indication, basically, if you know your target, yes, in infectious diseases, it can be applied.
Thank you, Monideepa.
You're welcome, Tuhina. So nice to speak to you after so many years.
Yeah, so nice to see you.
Okay, thank you, ma'am.
And I apologize if I went over time. Sorry about that.
Your
No
data
problem.
was so nice and so engaging that we don't complain.
No, if there are
Very
any
nice,
questions,
Monideepa. Very nice talk.
thank
After
you so
so
much.
long, I am seeing you. Very nice talk.
Thank you so much. And if there are any other questions, you can always email me, or we can address it. It's very great
Right.
to see you. Really great to see all of you. Yesterday, I tried to stay up, beyond 2 a.m. I couldn't. So I missed some talks. Sorry about that. And Tuina, I missed your talk.
No worries.
Thank you.
Okay. Thank you. Now Ashutosh will address the another session.
Yes, we'll be starting in 5 minutes.
I am Maza.